Hello, good morning, everyone. Welcome to this video in a very long time. However, I've been working on something, and that's something you see in front of you right now. I worked on graph neural networks for finding drugs for a very popular or very influential cancer target, that is HER2. So, our target is part of the ERBB family, and HER2 is a receptor kinase that is encoded by the ERBB2 gene. It is a well-established oncogenic target owing to its role in uncontrolled cell proliferation and resistance to apoptosis. So basically, it drives uncontrolled cell growth, and it's causing a signaling that is avoiding, or that in the end results that the cancer cells don't die, so they live longer, and so they can grow faster, and they can grow quicker. 20% of breast cancers are HER2 positive, so for this mutation. So for the method, I used a graph neural networks. They have emerged as a transformative deep learning technique for analyzing graph structured data, which is, I guess, obvious. These excel at capturing complex interdependencies within graphs, making them ideally suited for modeling protein-ligand interactions. So exactly what we are doing. And these networks have a really, unique advantage, and that is their message passing mechanism, which enables us to get really great feature extraction, and we can basically really understand the small parts that in the protein, as well as our not as much as in the protein, but also very more in the graph encoded data, and that is the possible drug compound. For the dataset, I used Kiba. That is a dataset consisting of 467 human kinase targets. They were tested with around 52,500 chemical compounds, and basically optimizing the label metric among them, and that are the Ki, Kd, and IC50 values. Basically, in layman's terms, the lower the better. The less drug you need to put into someone, that the drug is reactive, so to say. I'm also not a biology expert, as you can tell. Yeah, so the network, which is the most interesting part, the smile string is encoded into the graph, into a graph. That graph is one hot encoded and is then fed into a graph attention layer, fed into graph convolutions, and into global pooling, one-dimensional dense layers. At the same time, we have a separate branch that is a protein target sequence, which is basically converted to a numerical format in the embedding, fed into a bidirectional LSTM, and then fed into 1D convolutional layers, basically after that flattening them out and adding some more dense layers. These are then concatenated and then fed into multiple 1D dense layers as well. And in the end, we have the prediction of the protein binding affinity, or protein ligand binding affinity, and that is exactly what we want. And all activation functions are ReLU, just to include that. And the results were rather interesting. So the search space I expanded to binding DB, which is a database that scrapes patents, and we found some pretty interesting results. And these three were compounds that were previously developed by GSK for another kinase target, and the resulting affinity values for this one, and this one were 1.19, which is really good if it's actually true in the lab, and then 2.84 and 7.96, which is not really promising. Generally, as I state here, generally compounds under 5 are promising to further investigate in the lab. Why is that? Basically, low resulting values mean that the drug is potent at low concentrations and thus will show lower systemic toxicity when administered to the patient. Ergo, high binding affinity. So basically, the less drug you have to put into the patient, the better, and the more reactive it is with the intended target. So that is my quick video on what I've been working on. As I said, I'm not a biology expert, and I focus more on the machine learning part. And I hope you liked it. And if you liked it, give me a thumbs up and subscribe to my channel. And thank you very much.